case numbers we see? They're based on the PCR test. Just how reliable is it? SARS-CoV-2, like many other viruses, contains genetic material called RNA. RNA fragments are so small, they're difficult to detect. The PCR test doubles the fragments in what are called cycle thresholds and keeps doubling them until they have enough genetic material to identify. Most labs go up to 38 or 40 cycles, which means an amplification of 1 trillion times. But is that too much amplification? Because while live virus is present in people who are actually sick, traces of dead virus are present in people who used to be sick, even months later, or in people who were exposed to a virus, but their body cleared it and they never got sick at all. They're not infectious. Genuinely sick people get a positive test in relatively few cycles because they have a high viral load. But the samples of people who are recovered or exposed have to be magnified billions of times to get a positive result. That's why scientists are recommending not testing over 30 cycle thresholds. So what would happen to case numbers if labs cut back to 30? At one New York lab, it resulted in a 63% case reduction. When a Massachusetts lab did the same, almost 90% of their cases disappeared. And that's not the only way that PCR tests can go wrong. They can also misidentify remnants from the other 6 human coronavirus types as SARS-CoV-2.
And in fact, if you're testing in a population that doesn't have very much COVID, you'll get false positives almost half the time. The person actually doesn't have COVID, they have something else, they may have nothing.
In 2007, PCR tests caused a pseudo-epidemic for whooping cough at a hospital in New Hampshire. 100% of the results were false positives.
Because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body.
This is a delicate test that's supposed to be done by highly skilled technicians in a sterile lab, yet it's currently being done on a massive, expedited scale with hastily trained technicians working on huge numbers of tests. So the next time you see case numbers, ask yourself how many of them aren't cases at all.
Let's talk to Mike Eden. He's former chief scientific advisor with Pfizer, the big pharmaceutical companies. We've spoken to him, as you'll know, many times before about things like the false positive rate and the problems with mass testing. Now, this, of course, comes at a time when we're about to see the mass testing with a rapid test over pretty much the entire population of Liverpool. That is the latest plan. But is that the answer to the problem? Mike Eden, good morning to you.
Good morning,
Good
Julia.
morning. We've been, virtually everyone I've spoken to, whatever we're talking about, anything to do with lockdown, anything to do with the problems with the hospitals and the like, they constantly come back to it's the problem with testing and test and track and trace, and that we're not testing enough people, we're not testing enough of the right people, and we're not getting those test results back. Is that the problem?
No. Good morning, everybody. It's really, it sounds counterintuitive, but in my opinion, you don't chase respiratory viruses around the country using industrial molecular biology. I don't think we should be testing anybody unless they turn up to a hospital. And even then, we should be doing it better than we're doing. The test is not to be trusted. Everything they're telling you about, cases, admissions, people in ICU, and deaths, all pivot off this PCR test. People saying the R number. The R number depends on the PCR test, which I'm telling you is unreliable. More COVID patients going into hospital depends on the PCR test, which I'm saying is unreliable. Even the people operating it could not stand in front of you and say what percentage of the positives are genuine.
When we see these stark figures and they're reported in very sort of ponderous tones on the television, we've had 500 more deaths or 400, whatever it is on any given day.
Taking the total number across the UK to 41,759.
Often, actually, if you dig into it, you look at actually over the days in which they've actually been, the date of death as opposed to reported death, actually we're looking at far lower numbers. But also these are deaths with COVID, not of COVID. So you can be in hospital for a whole variety of other reasons. You can have heart disease, you can have cancer. If you test positive for COVID, you still end up on these statistics, don't you?
And see, this is what I'm saying. Everything you're being told, every scary fact and prediction from Whitty and Valence, all hangs off the PCR test, which they have admitted they cannot tell what percentage of positives are false and true.
When you do a PCR test, you basically amplify the amount of RNA, you put it into a DNA copy, and then you exponentially double that amount of DNA. and that's called the cycle threshold. And the more times you do that, the less virus you start out with. And this is important because you can basically pick up a single copy of RNA in an individual.
All right. There have been a number of reports of patients who shed viral RNA for weeks, as determined by PCR. It doesn't seem to be infectious virus. And the real question is, are they a threat for transmission? And I'm wondering if you think we could use a cutoff of viral loads determined by PCR to say this patient is no longer infectious, can go home, can go to a nursing facility, because right now the physicians are really having a hard time with that.
Right. Again, a good question. And what is now sort of evolving into a bit of a standard that if you get a cycle threshold of 35 or more, that the chances of it being replication competent are minuscule. Mm. And it's very frustrating for the patients as well as for the physicians. Somebody comes in and they repeat their PCR and it's like 37 cycle threshold. But you almost never can culture virus from a 37 threshold cycle. So I think if somebody does come in with 37, 38, even 36, you got to say, it's just dead nucleotides, period. Mm.
We have found RNA shedding in evidence for 78 days because the RNA is only 20 base pairs long and it takes much longer for you to degrade the RNA. So when you're picking up asymptomatic people, you have no idea if they have active infection or did they have it 2 months ago, because you intermittently shed this RNA. And it's the same for all viruses.
We know that, because it's been shown, that people who've recovered from the virus and they're clinically well, and you can no longer culture virus from a sample from their mouth or airways, still can be positive in the PCR test. And that's because, as you say, the test is not a test for virus, it's not a test for living virus. The test looks for a particular piece of the genetic code. And if it finds a small piece of a dead virus that's still stuck in your mucus and is being shed from cells, you could still be positive on a swab test for a long time.
So I think a large proportion of the so-called positives are in fact what I call cold positives. They're correctly identifying that there is some viral RNA in the sample, but it's from a dead virus. It can't hurt them. They're not going to get ill. They can't transmit it to anybody else, so they're not infectious. So that accounts for a large number of the so-called positive cases. These are people who've beaten the virus. Why are we using this test that cannot distinguish between active infection and people who've conquered the virus? This test has never been used in this way, and I've worked in this field. It's not a suitable technique. It's the kind of technique you would use for forensic purposes. If you were trying to do a DNA test to establish whether or not a person was at the scene of a crime, you would not be doing these tests by a windy supermarket car park in what looks like plastic marquee tents on picnic tables. It's not suitable at all, and it definitely shouldn't be done in the way it's being done.
The PCR test, PCR is a wonderful piece of science, but as its inventor, Nobel Prize winner Dr. Carrie Mullis pointed out, it was not designed on scale to be any kind of quantitative test. I know there are these pseudo qPCRs or quantitative PCR tests, but when you're doing 10,000 or 100,000 tests a day, as in UK, using amateurs, people who've never worked in a lab before, people who are so inexperienced at working in laboratories that they had 2 people to a flow hood, that they had to be trained how to use a micropipette, a Gilson pipette. I'd never seen a job called pipette trainer. A friend of mine who was a very experienced laboratory PCR technician for decades, worked in one of the largest UK private labs, the so-called super labs or lighthouse labs. And he couldn't believe it, that on his first day he was introduced to someone whose job was pipette trainer. And he asked her, just to show me what training he would provide. Obviously, he knew how to use a pipette.
And it was obvious in the first 30 seconds she had no idea how to use a Gilson micropipette. And at coffee, he asked her what previous jobs she had had before coming to this very important role. And the answer was supermarket shelf stacker at night. So that's the contempt with which the private companies that are operating on scale the most important diagnostic test ever in the history of the UK, that's the contempt with which management is choosing its staff. So even if PCR, if done well, might distinguish an infected person, from clinically infected person from a not clinically infected person, in practice, forget about it. They've got graduates who have usually got a science degree but no practical commercial lab experience. They're being supervised by this one example, a person whose last job was stacking shelves in a supermarket and ostensibly training them. And just for a moment, contamination is a major problem in PCR. It's an amplification technique, and at high numbers of cycles, Rainer, as you said, theoretically, you might amplify what was in the sample by 1 trillion fold. So it doesn't take much imagination to believe that a few particles floating in the air could contaminate an entire rack of open tubes, notwithstanding a flow hood. And if you have poor pipetting technique, you easily generate aerosols every single time you empty the pipette tip by depressing the thumb. So the person I spoke to who's done this for decades said, I asked him what he thought the operational false positive rate arising purely from contamination, cross-contamination. I asked him what to estimate a percentage. He said it could be 0.2, it could be 2%, it could be 20%, it could be any number at all. And I asked him, well, presumably they run no negative controls through the entire chain of custody. He said, no, they don't do that. They don't do any negative controls. They just add a plate control right at the end before it goes in the PCR machine. So it's pure and simple fraud, in my country at least, absolute fraud. I don't believe a single set of statistics that arise from UK mass-scale PCR testing. That's what I think.
So it's very important that the PCR test is only applied in those acutely sick. And the regulatory bodies in the United States have never approved the use of the PCR test or the antigen test for that matter in people with no symptoms. Never. And the reason being is because if people don't have symptoms and we do these tests, they're more likely to generate a false positive test than to generate a true positive test. So they are only approved as a diagnostic aid in an acutely sick individual. And under no circumstances should testing be done on asymptomatic individuals. So that means no weekly testing, no punitive testing. There's testing people getting on airplanes. None of that is indicated or clear by any regulatory agency. Don't do it anymore. So if anyone listening here is in a leadership role and you're in a school or somewhere else and you're trying to make a decision about testing, just use testing in acutely sick individuals, typically done by health professionals.
Until today, the most panic-inducing principle, namely that most media still speak about today we had so many new infections.
Well, the latest government figures show the highest daily number of new infections.
That's the increase in the number of daily infections.
With coronavirus infections in England doubling every 7 to 8 days.
The rate of infections are growing significantly.
Yesterday, you might remember, we reported the highest number of infections. The figures out today have shown an uptick in infection rates in London.
A steep rise in coronavirus infections in England.
322 new cases of infection.
With what is clearly a second wave of infection about to break.
Which is it's not true. A virus, it's a particle. It goes to everybody. So if somebody is immune, the virus also goes into this body and it will multiply even a little bit. But if you're immune, you attack the virus first with antibodies and you make immediately debris. The virus is destroyed. You have only parts of the virus around in your tissue. If you make a swab, or in your blood, if you go and test you will find this debris, and part of the debris will be nucleic acids, RNA. And if you make a PCR, all these people will be positive because the PCR picks up 2 tiny, or if it's a good assay, 3, sometimes 2, sometimes only 1 tiny little piece of RNA that is then amplified. And this assay cannot tell you whether you had the virus or you just had some dead chunk of the virus, which still gives you a positive result. So corona positive with a PCR, it tells you only a little tiny bit of DNA, of RNA, nucleic acid was there, but it doesn't tell you whether it was what the virus complete or even a virulent, meaning a virus that can really make you sick. All this is not in this test. No other virus ever on this planet has been accompanied by so much testing, and a testing that has created so much nonsense and panic.
So is the threshold cycle is reporting out a pretty standard practice in doing a diagnosis now rather than just positive or negative? Okay, good.
Yeah, I mean, if when you go in, when I get my test, it's negative. When someone comes in and it's positive, they don't give them the threshold until you go back and ask for it.
Okay, but they've got it.
They've got it.
All right.
They've got it.
How did we get here? Why are we even using this test? So in January, a German virologist named Christian Drosten, he created a new laboratory assay to detect the coronavirus from the German Center for Infection Research, and he put out the WHO quickly picked that up, picked up that guideline, and the Euro Surveillance also published that in their journal. It's a infectious disease epidemiology journal, and it's called Detection of 2019 Novel Coronavirus by Real-Time RT-PCR, and that was in January. A lot of people had an issue with that. They took to Twitter. A guy named Walter Akima went on Twitter and really questioned why the heck that thing was rushed so fast. He basically said, of all the publications, no research paper got reviewed and accepted in less than 20 days. One got accepted in less than 24 hours after submission, and that was the Drosten paper. It's come to be known as the Drosten Review. So now the breaking news on this is this paper is being contested, as is and by extension, so is this new assay PCR test that is being used, and that's the PCR test that's being used really everywhere throughout the world.
This is what's driving the lockdown, right? I just saw, California, you're not allowed out of your house. You can't get in a car. You can't get on a bicycle. You can't do anything because the PCR tests are showing that there's this dramatic increase in cases.
Yeah, so the PCR is being contested by 22. These are credible health professionals. They've done an external peer review because they're basically stating the first one was not peer reviewed. They sent it to Euro Surveillance editorial board asking for a retraction. What they did was looked at all components of the test design. The protocol recommendations were assessed. They looked at the parameters and they examined those against relevant scientific literature. And now this is a deep dive, so I recommend anybody go into this, but I took some segments from it, and let's just read a couple here just to give you an idea what these tests, some of the issues with this test. So, and there's 10 of them. There's 10 major errors in there. I just took a couple out. Number 3, the test cannot discriminate between the whole virus and viral fragments. Number 6, the PCR product have not been validated at the molecular level. Number 7, the PCR test contains neither a unique positive control to evaluate its specificity for SARS-CoV-2 nor a negative control to exclude the presence of other coronaviruses. Number 8, the test design in the Corman-Drosten paper is so vague and flawed that one can go in dozens of different directions. Nothing is standardized, and there is no standard operating procedure. And number 10, we find severe conflicts of interest for at least 4 authors, in addition to the fact that 2 of the authors of the Corman-Drosten paper, Christian Drosten and Chantal Ruskin, are members of the editorial board of Euro Surveillance, which may explain why it was peer-reviewed so quickly or put in that journal so quickly. Now, this is something that's very interesting, and it speaks to the cycling. And this is directly from the paper. We've talked about the cycling. They go right in. This is probably the most concise description I've ever seen. And they say about the cycling, if someone is tested by PCR as positive when a threshold of 35 cycles or higher is used, as is the case in most laboratories in Europe and the US, the probability that said person is actually infected is less than 3%.
Whoa.
The probability that said result is false positive is 97%.
My God. oh, we're destroying the world with this test and it's 97% inaccurate. Did you hear that, folks? This is not me. This is not somebody, a reporter out there. This is a pretty large group of scientists involved in this too, right? Authors on this rebuke of the PCR test.
Yeah, there's 22, and these are not run-of-the-mill scientists. These are researchers. These are world-renowned doctors and academics, people that have held positions in pharmaceutical companies such as Pfizer. Now, let's look at the conclusion of this paper. I know it's a long segment, but it's so important. So they say, in light of our re-examination of the test protocol to identify SARS-CoV-2 described in the Corman-Drosten paper, we have identified concerning errors and inherent fallacies which render the SARS-CoV-2 PCR test useless. If there's a headline, that's it right there. And finally, they say a decision to recognize the errors apparent in the Corman-Drosten paper has the benefit to greatly minimize human costs and suffering going forward. Is it not in the best interest of Eurosurveillance to retract this paper? Our conclusion is clear. In the face of all the tremendous PCR protocol design flaws and errors described here, we conclude there is not much of a choice left in the framework of scientific integrity and responsibility.
Boom. Wow, Jeffrey, that's gigantic. That's gigantic. We have, and this really is behind this entire pandemic. Without the false positives of this PCR test, none of this story adds up.
So the test that we have been using for diagnosis looks for RNA, which is a viral equivalent of DNA. It's what it uses to replicate. And that test can fail. And it can fail when you put labs under pressure, when you're doing high volume, high speed testing, then you can end up with cross-contamination, but also all sorts of other problems. And it's well described in the medical literature that with PCR testing particularly, you can get false positive pseudo-epidemics. And I think that's what's happening here.
Well, the major problem is that the PCR test, whenever it's positive, and I told you before that this test is worthless because it's falsely positive to a tremendous extent if you go around testing normal and healthy people. It should be prohibited. It's treacherous. The treachery is that a normal person with a positive PCR test is graded as a COVID-19 case, which means that he's graded as a seriously ill individual. And he enters the records as such. And this must be stopped because it inflates the numbers to an unbearable extent and to an unethical extent. That's what's happening today. The PCR tests are totally inaccurate because they're being conducted in a non-standardized manner. You know, the PCR method developed by Mullis, who got the Nobel Prize for this, he himself said, do not use this test for diagnosis. And in fact, neither was it developed for diagnosis, nor has it ever been approved for diagnosis. At best, this test may be used to support the diagnosis, the clinical diagnosis, meaning that you should not be using this test on people without symptoms, as it's being done right now. This is abuse of a test. Why? Because the test is far, far too sensitive. You know, last week, 60 people were sent to quarantine in Bavaria, in a town. And they decided to demand that the test was repeated because they didn't believe it. And the test was repeated and it turned out that 58 were negative. And the last 2 were also only positive because the test had been rigged so that it was so sensitive. But the other 58 were totally negative, meaning that the test itself was faulty. So there you are. In fact, this test should be trashed immediately, worldwide. And it should be considered a criminal act for anyone to be sent to quarantine because this test was positive.
I think it's not unreasonable to say a best guess of the false positive rate at the moment, what's called the operational false positive rate, is about 5%. 5% of 300,000 is 15,000 positives. I think some of those positives are real. I don't think it's very many. Now, the problem with this false positive issue, it doesn't just stop at, quotes, cases. It extends to people who are unwell and go to hospital. So people who go to hospital having tested positive, and it could be a false positive, and I think most of them are at the moment. If you go to hospital and you've tested positive previously, or you test positive in hospital, you'll be counted now as a COVID admission. But I think you know where this is going now. If you should now die, you'll be counted as a COVID death. But that's not correct. These are people who might have gone to hospital having had a broken leg, for example, but 3% of them will still test positive, and they haven't got the virus. It's a false positive. And if they die, they'll be called a COVID death, and they are not. They've died of something.